In this module, we will discuss another important technique which is used in routine by molecular biologists to determine and to characterize the nucleic acid and the protein. The technique is known as electrophoretic technique. That is the electrophoresis of these molecules. Now, the general principle of this technique is that there is a migration of charged particle under the influence of an electric field. That is when the particles, they are subjected to an electric field, they migrate toward different poles. And the movement of charged particles under the influence of an electric field, that is anion and the cation, positive and negative, that will migrate to the respective electrode. As the similar charged molecule, they repel, whereas opposite molecule, they attract. So using this principle, the electrophoresis technique is used. Now, the equipment which is required for electrophoresis, it consists basically of two items. One is the power pack, that is which supplies the electric current or the electric field, and the second one is the electrophoresis unit, that is a unit which contains our molecule, our particle, which we want to study that where they are migrating to. Now, electrophoresis units, they are generally designed for running either vertical or horizontal gel system. That is, either they can run vertical or horizontal gel, and the gel is actually used to support the particles, that is those particles which are there and they can migrate. Now, vertical slab gel units are routinely used to separate protein in acrylamide gel. So you see, there are vertical as well as the horizontal gels. Verticals are generally used to separate the protein in acrylamide gel. And in this picture, the most commonly used mini gel apparatus is being shown, where you can see that power supply and the gels which is being charged in a tank which contains the buffer or the media and all that, and then it runs on the gel there. Now, generally the gel dimensions, that is what is the size of the gel, they are typically 8.5 centimeter wide, 5 centimeter high, and 0.51 millimeter, that is a half centimeter thick. That is the gel which is used to run the electrophoresis. Now, when the apparatus is assembled, the lower electrophoresis tank buffer, it is surround the gel plate and afford some cooling of the gel plates, because the gels are there and there is a buffer which acts as a connection between the power supply and all that, and at the same time it helps to cool the gel plates which are there. In this slide, a typical horizontal electrophoresis apparatus is being shown, where you can see that there is a gel, and gel is connected by the wick of paper into the two buffer reservoirs on the left and the right. And in the reservoir, there is electrodes, one is positive and one is negative. The gel is placed on a cooling plate, and the electric current, it flows through the cooling plate and helps to migrate the particle toward their respective charges. Now, connection between the gel and electrode buffer, as is seen, is made using a thick wad of wetted filter paper. That the wetted filter paper are placed and the gels are connected. The power pack, it supplies a direct current between the electrodes in the electrophoresis unit and the particles. They move to the under this electrical field. Now, when a potential difference is applied from the power supply, molecules with different overall charges, they will begin to separate owing to their different electrophoretic mobilities. That is the basic principle of this, that under a potential difference, the molecules, they start to move according to their different electrophoretic mobilities, their size, their charges, and all that. And the current in solution between the electrode is conducted mainly by the buffer ions. The buffer ion which are provided, the current is conducted through that because it contains ions. Now, electrophoretic medium, definitely when a potential is applied, it gets heated during its run. And heating of electrophoretic medium, it affects the electrophoresis in the following manners. Number one, that an increased rate of diffusion of sample and the buffer ion. When the temperature is heated, an increased rate of diffusion is there and of the sample in the buffer ions. The formation of convection currents, because when electric current is passed through that, it is heated, the convection currents, they are made in the buffer. And then the thermal instability of the sample, that are rather sensitive to heat. We have to consider this, that the molecules, they are sensitive to heat and they shouldn't be thermally unstable, that they shouldn't break under that heat. And the last is that the buffer, when it is heated, its viscosity is decreased. That is, it becomes less condensed. It becomes less viscous. And with that, it affects the electrophoresis of the molecules. But in certain cases, the heating, it helps it as well. And the separation of molecules can be performed better. It can be better and make the good analysis of different proteins which are present there according to their charges on them. So as a matter of fact, we see that this technique, electrophoresis, helps us to separate the molecules according to their size, according to their potential, and it gives a very useful handle, a useful technique to the molecular biologists for studying the proteins or the DNA and all that.